Hello pharma aspirants, welcome to online GDC classes. So in our new series, that is nothing but the Target Pharma 360 degrees, we have already started the MCQ series. Okay, so in this Target Pharma 360 degrees, we are going to discuss about all types of the MCQs relevant to all the subjects in our pharmacy course, which are going to be helpful for you in cracking the competitive exams like drug inspector, pharmacist, GPAT, NIPER, all these things. Okay, so let us start our new series with the topic UV spectroscopy. Already we have given two parts related to the UV spectroscopy in this Target Pharma 360 degrees. Okay, part 1, part 2 were already uploaded in our GDC English channel. Most of you have already viewed those topics also. So this is a part 3 which is related to the MCQs of UV spectroscopy subject. Okay, so let us go into the questions now. The first question, if you see in today's session, that is, in which of the following ways absorption is related to the transmittance? Okay, so absorption and transmittance, how these two are interrelated. We know that absorbance, whatever it is there, we are going to get it from the spectrophotometer. Okay, so that is nothing but the formula log of percentage T to the base 10. So in this way, absorbance and transmittance are going to be interrelated. Okay, so actually the spectrophotometer, whatever we are going to use in case of the UV and visible, it is going to measure the transmittance only. So that transmittance, it is going to be converted into the absorbance, because it is an absorption spectroscopy. The molecule it is going to be under analysis is absorbing the light. Okay, but whatever the transmitted light it is there, that is reaching the detector. That's why the formula it is required in order to convert in terms of the absorbance. So the absorption which is related to the transmittance. So let us see, absorption is a logarithm of the transmittance. Absorption is the reciprocal of the transmittance. No, not at all. Absorption is a negative logarithm of the transmittance. That means minus log of this one. Somehow it is going to be that. And absorption is a multiple of the transmittance. No. So A and C, if you observe, absorption is a logarithm of the transmittance, but negative logarithm if you are going to observe this in this case. So obviously C is going to be the correct relation. Okay, so 2 minus it is there, that is okay. So because log of 100% transmittance, log 100 is going to be 2 value. So 2 minus 2, absorbance will be 0. 100% transmittance has been observed means there is no absorption in the molecule. Okay, so that's what the formula is indicating. So absorption is a negative logarithm of the transmittance. So this is the right option. Next question, the representation of Beers-Lambert's law. Okay, so in the UV spectroscopy, Beers-Lambert's law is more important, which is given as A equal to ABC. In simple terms, we can easily remember this Beers-Lambert's law. Okay, A equal to ABC. So in this, if you are going to see, that means the formula whatever we can remember very easily, A equal to ABC. So in this, B represents the distance. Actually, it is a path length. Okay, and C represents the concentration and A represents the absorption. Then what does the small a represents? So when we are going to use this small a, let us see the options. Intensity, no. Transmittance, no. Absorptivity and admittance, this is irrelevant completely. Only the word absorptivity. Actually, whenever you are going to see in most of the textbooks, the formula epsilon into L into C. Okay, so epsilon it is going to be called it as molar absorptivity. So this molar absorptivity is going to be very high for the highly intense absorption bands. Okay, and L is going to be considered as the path length and C is the concentration. Okay, so when this formula is becoming A equal to ABC, in the simple terminology we want to remember in this case means whenever B is going to be considered as the same as that of the L, path length, C is the concentration, A is the absorbance, and this small a it is going to be considered as the specific absorbance value. This specific absorbance means whenever this absorbance it is going to be considered as 1% and 1 cm. Okay, that means A equal to ABC formula it is there. That means you are going to see path length it is going to be 1 centimeter and concentration of the solution is 1. Okay, so in that case what happened, A into 1 into 1. So capital A is becoming equal to the small a. So in that case, the absorbance and specific absorbance both are equal. That's why we are going to call them as absorptivity value, specific absorbance, or it is going to be generally epsilon value. If you are taking means it is called as molar absorptivity. Okay. So let us move to the next question. Transmittance is given as, transmittance formula they have given T equal to P by P naught. That is the P naught, it is going to be considered as the power of incident light on the sample. Power of the incident light, and some of the textbook you are also going to see it is I by I naught also, right? And what does P represent? So I by I naught, so this 0 always it is going to be representing the incident light radiation, okay, and simple I means it is a transmitted light radiation. So similarly here also P by P naught means power which is related to the incident light, which is normal P, which is related to the transmitted light. Okay. So now let us answer this question. Small p, they have asked the P, not the P naught. P naught means the incident light radiation power. Now P means that is a transmitted light radiation power. So let us see the options. Radiant power transmitted by the, okay. So obviously this is going to be the right answer. Radiant power absorbed, no. Some of the powers absorbed, no. No, not at all. The D option is also right. So only the option that is A is going to be the right answer. Okay. Next, we will move to the fourth question. Which radiation source has electrode in its construction? Okay. So the lamps we are going to use in the spectroscopy, different types of the lamps are going to be used. For UV, deuterium lamp and mercury lamp also is it is suitable. And fluorescence specifically we are going to use a mercury lamp, and for visible means tungsten lamp. So in this they have asked in the source itself an electrode is present. So which type of the source that is? Tungsten lamp, hydrogen lamp, xenon lamp or mercury lamp? Which type of the lamp we have an electrode? And in the given options if you see hydrogen discharge lamp and hydrogen electrode, it is going to be there. So in that case you are going to see the correct option, that is B, hydrogen lamp. Okay. Tungsten lamp means a tungsten element, it is going to be there. It is a small filament like our household bulbs we are going to use. And xenon discharge lamp means xenon gas is going to be there. Mercury lamp means mercury, it is going to be filled up here there. Okay. Only the electrode hydrogen gas is going to be used, and in that it is going to be electrode. Okay. So an electrode, it is going to be fixed in that. So standard hydrogen electrode. So in the potentiometry also you might have heard about that one. Okay. So let us move to the next question. Photovoltaic cells is also known as, okay, photovoltaic cell is nothing but a detector in the UV spectroscopy, and in this let us see the options. It is also known as phototube, photoemissive tube, photomultiplier tube and barrier layer cells. Okay. So we know that photovoltaic cells which are going to be used as detectors in the UV spectroscopy. Not only photovoltaic cells, phototubes are also used, photoemissive also, all these are the UV detectors. Okay. But the other name for this photovoltaic cells is barrier layer cell. How this photovoltaic cell it is going to be there? You are going to see a superconducting material, you are going to semiconducting, not superconducting, okay, semiconducting material you are going to see on the top of this. So above and below you are going to observe the semiconductor materials. So in between that you are going to see the selenium layer, it is going to be present. Okay. So thereby you are going to see the whenever the light rays, so light rays it reaches this surface. Okay. So anti-reflective glass they are going to use here, and semiconducting material whatever like silver, gold, like that they are going to use, and another semiconducting material like copper generally they are going to use. Okay. So photovoltaic cells, these are also called as barrier layer cells, means here you are going to see the two metals layers are going to be interconnected with the selenium layer. So thereby whenever light hits on the surface of this cell, you are going to see the passage of the electric current. Okay, so it is going to be connected to a voltmeter, so that you are going to see the production of the electricity. Okay, so like a solar cell, how we are going to use for the generation of the electric current. So that is also nothing but a photovoltaic cell only. So this is also called as barrier layer cells. Okay, so let us move to the next question. The detectors used in the UV visible spectrophotometers are also called as, generally detectors, whatever the previous slide we have seen, all those are the UV detectors only, photo tubes, photo multipliers. Okay, and these are called as photometric detectors, photovoltaic, photoemissive cells, photo multiplier tubes. Actually the name of all those detectors, photo multiplier tubes are also UV detectors, photoemissive cells, photovoltaic cells, all these are also UV detectors. But what are those called as? Actually photometric detectors. Okay, so the right option it is A. Next question, in photovoltaic cell which unit act as an electrode? Okay, so that means what is the, already we have seen two semi-conducting materials are going to be there, and those are going to be interconnected with the selenium. Okay, so in which electrode, which one it is going to be acting as those electrode means the metallic layers, or those two metallic layers are going to be acting as electrode. So option B it is going to be the right answer. Plastic case, glass, no, these are not the right options. Next, the preferred source of light in the UV spectrophotometer. Okay, that means what is the source of radiation generally we are going to use for the UV radiation. UV radiation, what is the wavelength? 200 to 400 nanometers, right? So for this, whatever the different sources are also available, but what is the preferred one they are asking. Here what are the options they have given? Tungsten lamp, no, this is not at all a UV radiation generating source. It is going to give you the visible source. Hydrogen discharge lamp, mercury lamp, deuterium lamp. Okay, so this can also be used as the UV and both the UV and visible, but there are some drawbacks in the UV region, so that's why we are not going to use this. And deuterium lamp, that means D2 lamp. So deuterium is isotope of the hydrogen. This one it is going to generate the UV radiation in the range of 200 to 400 nanometer. So that's why it is the highly preferred one. Okay, so D option it is going to be the right answer. Next, which of the following solvent is commonly used in the UV spectrophotometer? So solvents for solubility of the analytes, whatever we are going to take the spectrum in the UV and visible regions, what are the different types of the solvents we are going to use. But whatever the solvent it is, you should know about the UV cutoff. That means the wavelength above which only those solvents will not have any absorbance. Okay, so UV cutoff point, generally we are talking about UV cutoff means this is represented for so many solvents, cutoff point. Okay, so cutoff marks, you are well aware of those terms. Cutoff point means UV cutoff means the wavelength at which it is having some absorbance value until that. Okay, so if you are taking about the water is UV cutoff point is 190 nanometer. That means if you are going to use the water as the solvent in the particular analysis by UV, above 190 nanometer only you have to analyze the particular analyte. So if you are in and around 190 means it is not a suitable method. You need to change the solvent again, right? Because at that 190 nanometer, water is also giving the absorbance value. So it is an interference that is not acceptable, right? So that is the UV cutoff point. So here they have given water, chloroform, methanol and all of the above. That means what are the different types of the solvents? Solvent generally they are asking means all these solvents are going to be preferred in the UV spectrophotometer, but depending on the UV cutoff point you need to select that. Okay, so option D it is going to be the right answer. Next question, if you see, in UV spectroscopy the sample cells are made up of. Okay, so in the instrumentation part if you observe, so from the source of light radiation, the radiation is going to be first enters the monochromator, then the polychromatic light converts to the monochromatic light, then it enters the sample cell. So those sample cells are going to be used, which are of different path length, it is also going to be there, as well as you are going to see them in the cuvettes. Actually those samples here we are going to call them as cuvettes. Okay, so these cuvettes are going to be at different capacities also. So one side smooth surface and another side rough surface will be there. So on the rough surface only you have to hold it, the smooth surface, the light radiation, it is going to pass through that. Okay, and these cells, these sample cells are going to be made up of which material? They are asking, either glass, quartz, polystyrene, metal. Okay, so especially for the UV spectroscopy, quartz is a material. Quartz is also a glass, actually one type of the glass. Okay, but normal glass, it is glass is again 4 types. Okay, that is a different thing. So one type of the glass material, that is a quartz glass, we are going to use for the sample preparation, for the sample cells. Okay, so as a cuvettes we are going to use, the cuvettes are made up of quartz material. So those quartz material cuvettes only we have to use it for the UV spectroscopy. Otherwise what happens? So this quartz material, whatever we are going to use for this manufacturing of the cuvette, it is allowing the UV radiation without any absorbance. Okay, so if you are going to use the glass cuvettes, normal glass, whatever we are going to use it in our household purpose, whatever the glass tumblers you are going to see, otherwise in our cars and houses also you are going to see some of the cars. Okay, that glass, it is going to absorb the UV light. That means it is not allowing the UV light outside, so it is not going to pass through that. Okay, so that's why these are not going to be preferred. Quartz will allow the passage of the UV light. Okay, it is not going to absorb that UV light, so that's why it is a preferred one. Next question, the gratings in the UV spectrophotometer. Okay, so here also the same thing, whatever the dispersive elements you are going to use in the monochromators. Okay, so in the olden days they have used the filters, afterwards it become the prisms, then now it is gratings are taking a lead. That means highly dispersive elements these are. So these gratings are also made up of quartz only. Okay, so glass, polystyrene, potassium bromide, no potassium bromide, the cells are made up of those potassium bromide are going to be used in the IR spectroscopy, not the UV spectroscopy. Okay, so the right option here also, that is C, quartz only. Next question, the most widely used detector in the UV spectrophotometer. Okay, widely used detector. So all the photo tubes, photomultiplier tubes, okay, diodes, all are going to be used as detectors in the UV, but widely used detector. Let us see the options first. Photo cells, barrier layer cells, 𝘂𝙎 cells and photomultiplier cells. Actually if you see the options here, in this they have given photo cells and barrier layer cells, photomultiplier. These three are related to the UV spectroscopy. 𝘂𝙎 cell, it is different. Okay, this is going to be used in the IR spectroscopy. So the preferred one, so which the most widely used detector, that is nothing but the photomultipliers. Okay, photomultipliers means it is going to multiply the signals, because there you are going to see diodes are going to be present there. Okay, so that the secondary electrons are going to be produced in case of this photomultiplier, and signal amplification it is going to take with less quantity or very minute quantity of the sample, it is also there, you are going to see the signal for that. Okay, so that's why photomultipliers are going to be the preferred detector in the UV spectrophotometer. Next question, in visible spectroscopy, if the color of the solution is red, then which color filter is preferred? Okay, so in case of the filters, whenever we are going to see the filters used for the UV spectrophotometer, so there are absorption is going to be a different thing. Okay, that means if you are going to see one orange color filter, okay, so it is going to absorb the another color. What is that? It is a blue color. Okay, so by looking wise you are going to see it is an orange filter, so that means orange color you are going to see, but generally what it is going to do, it is absorbing the blue light. Okay, it won't allow the blue light outside of this. So there are some colors which are absorbing the complementary colors. Colors and complementary colors, if you know, then you can answer this question. So for this purpose, let us first have a diagram related to the colors, primary colors and complementary colors. Okay, so we know with GR it is there. Okay, so that is the fundamental thing in the UV spectroscopy, visible spectroscopy. So in this, if you are going to observe the colors sequentially, if you are going to see, okay, so let us say a circle we can draw. So in this, we are going to observe means in this, that means primary color will be different and complementary color will be different. Okay, so if the primary color it is yellow, you are going to see the complementary color, that is nothing but the purple color. Okay, so if the primary color is red, then you are going to see the green color as the complementary color, vice versa you are going to see. Okay, so if the primary color is orange, you are going to see the complementary color, that is blue color. Okay, blue, indigo, all these comes in the same way. Okay, so if you remember these four, then it will be easy for you, right? So with GR we know violet, indigo, blue. So all these comes here in this region. Okay, so then purple, red, these are comes under above 500 nanometer. Okay, so orange, red, this yellow, orange, red. Okay, so it will start like this, the cycle, it will start like this. So if you remember the cycle in this way, it will be easy for you to remember to answer the questions. Okay, colors and complementary colors. Orange, if you are going to see, if it is a primary color, the complementary color it is going to be the blue color. Okay, so if the green color and red color, these two are primary and complementary. For vision purpose, you are going to see that is a red filter, but what it is going to do, it is absorbing the green color. It is absorbing, it won't allow green color outside of that filter. So the red filters are going to absorb the green color. Okay, so that's why you are going to see the complementary color. Those are called as actually complementary colors. Okay, and these are in case of the filters. So because filters are also going to be used as monochromators. Okay, not the actual monochromators, but the purpose it is going to be taking place with the help of the filters. Okay, so this diagram will help you in selecting the primary color and complementary color. Okay, so let us move to the next question. Deuterium discharge lamp is used as source. Very simple question. Deuterium D2 lamp, right? So D2 lamps are going to be used in the UV spectroscopy. Let us see the options: fluorescence, polarimeter, UV, IR. So definitely the C, it is going to be the right answer. Next question, if you observe, Woodward-Fieser rule is used to predict. Very well known question this is. Woodward-Fieser rule, which is going to be used for calculation of the lambda max for conjugated dienes as well as trienes, polyenes and alpha beta unsaturated hydrocarbons, acyclic conjugated dienes. Okay, and not only that, it is also going to be used for the bicyclic compounds also, right? And not only Woodward-Fieser rule, you are also going to see the Kuhn rule also, it is there. So this is also going to be obtained when the conjugation is more than 4. So Woodward-Fieser rule is applicable only up to the conjugation it is 4. So if there are more conjugated bonds are there, more than 4, then Woodward-Fieser rule is not applicable. Okay, so in that case you have to go for the again Kuhn rule, it is also available, and all these rules are going to be helpful in calculating the lambda max. Okay, so that is going to be the right answer. Next we will move to the another question. B band is found in which type of the compound? Okay, the question related to the absorption bands. So in the UV spectroscopy part 2 also we have discussed about these absorption bands. So if you want to know more clarification about the absorption bands, you can watch those videos also in our Target Pharma 360 as well as Pharma Thunderstorm series is also there, in which also we have this information related to the absorption bands of UV spectroscopy. So you can watch such videos in our GDC English channel for more clarification. Okay. So B band is found in which type of the compounds? Options: aromatic, cyclic, aliphatic and alcoholic. So actually if you are going to know about the types of the bands in the UV spectroscopy, means there are 4 types of the bands. Okay. So alphabetically if you want to remember, means B band it is there, E band, K band and R band. So there are 4 types of the absorption bands, but R bands are actually forbidden transitions. So mostly you are not going to see this type of the R bands. Most intense bands are K bands you are going to observe, and these B bands you are going to see in most of the aromatic and heteroaromatic compounds. Okay. So in these 2 compounds you are going to observe this B type of the absorption bands apart from the transitions. So in these 3 absorption bands you are going to see in case of the pi to pi star transitions, whereas R type of the absorption band you are going to see in the N to pi star transitions. Okay. So especially B bands they have asked in this question. So aromatic and heteroaromatic compounds you are going to see the B bands. Okay. And next we will move to the another question, that is 17th question, the change of lambda max. Okay. So lambda max towards the shorter wavelength it is known as. Okay. So whether due to an oxochrome effect or solvent effect, you are going to see the shifting of the lambda max either towards the shorter wavelength or longer wavelength, and in this if you are going to see the question, that is lambda max towards the shorter wavelength. Okay. So what they have asked, that is towards this side. So shorter wavelength, then what is this, it is called as, so wavelength is decreasing. So shorter wavelengths will give the blue color. So in that way you can remember, longer wavelengths are going to be red color. So if it is shifting towards the longer wavelengths, means that is called as red shift. If it is shifting towards the shorter wavelength, means it is called as blue shift. Okay. So in that way it is easy to remember, and red shift it is also called as bathochromic shift, and blue shift it is also called as hypsochromic shift. Here just they have given only colors wise. So shorter wavelength means blue shift, it is going to be the right answer. Okay. Next question 18th one, identify the correct statement. Okay, which statement it is correct? Let us see. The absorbance is intensive property of the substance when absorptivity is extensive. Molar absorptivity. Actually whenever we will give the light to any homogeneous solution as an energy, then only it is going to take up that energy. Okay, then only you are going to absorbance, it is going to be observed. So absorbance it is not an intensive property, and absorptivity it is not an extensive property. Actually they have reversely they have given in this option A. Let us see the option B. Absorbance is extensive property, this is right, and whereas absorptivity is the intensive property. So perfectly option B it is going to be the right one. Let us see the another options also. Both absorbance and absorptivity are intensive. No, not at all. Only absorptivity is the intensive property. Both absorbance and absorptivity are extensive. No. So obviously B option it is going to be the right answer. Okay. Absorbance is extensive property, whereas molar absorptivity it is going to be the intensive property. Okay. So we will move to the next question. Bonding and anti-bonding orbitals are. Okay. So whenever we will give light in terms of the energy to any molecule, so absorption spectroscopy, that is UV, visible fluorescence, IR, all these are there. You are going to see the bands, absorption bands are going to be observed. All these are going to be molecular level. Okay. Atomic absorption, atomic emission, that is going to be at the atomic level, but UV visible colorimetry and fluorimetry, IR, all these are at the molecular orbitals, these are going to happen. So bonding and anti-bonding orbitals are related to the molecular orbitals. Let us see the molecular orbitals and having the same energy. Okay, they are asking in terms of the energy actually. So here they are asking molecular bonding and anti-bonding orbitals are molecular orbitals only. They are having same energy. No, not at all. Atomic orbitals are having the same energy. These are not at all atomic orbital. So option B is nowhere correct. Molecular orbitals with anti-bonding orbitals are having higher energy. Yes, anti-bonding orbitals have the higher energy. Okay, so option C is going to be the right answer. Atomic orbital, no, nowhere it is atomic. So no need to again read this option. Okay. So bonding orbital, whenever the excitation it is going to takes place from ground state to the excited state. So in the ground state means normal energy. Okay, energy it is going to be quite normal. Whenever we will give energy in the form of light means from the ground state excitation it is going to takes place. So that's why it is called as highest occupied molecular orbital, and antibonding orbitals are lowest unoccupied molecular orbitals. Okay, which have the highest energy. Antibonding orbitals, that means pi to pi star. Always pi star is going to have the highest energy than pi. Okay, so option C is going to be the right answer. Let us move to the last question. Which of the following solvent has cutoff value below 200? Okay, so you should aware of the UV cutoff points to answer this question. So many solvents are going to be used in the UV spectroscopy, and most of the not all the solvents, very few, 10 to 15 number of solvents are there which have the UV cutoff point, like water, chloroform, acetone, carbon tetrachloride, okay, methanol. It is also having the UV cutoff point, but mostly we are going to use the near IR region in the range of 200 to 400 nanometer, right? So in this 200 to 400 nanometers means the solvents, mostly water and methanol. Water, it is going to be having the absorbance at 190 nanometer. Okay, so UV cutoff point. So that's why below 200 nanometer they have asked, means water is having the UV cutoff point, and another whatever the solvents we are going to use, like methanol also we are going to use, which is having a 205 nanometer. Okay, and acetone is having the absorbance at the 330 nanometer also there, and carbon tetrachloride is also going to have some absorbance in and around 210 nanometer. Okay, and chloroform is also going to have the absorbance in and around 220 to 240. Okay, so like that you are going to see the UV absorbance values, so in those regions. That's why UV cutoff point you should know about the solvent. Okay, so for this question water is the right answer. So these are the some of the MCQs we have discussed for the UV spectroscopy part 3. Okay, so for more such videos you can subscribe to our GDC English channel and get the more knowledge about the all the competitive exams related questions in your pharmacy course. Okay, so we will meet in the next session. Thank you students. Thank you for attending.